We have the pleasure to interview David Rahn, Professor of Cellular Pathophysiology and Clinical Biochemistry and Wellcome Trust Principal Research Fellow at the University of Cambridge in the United Kingdom. Professor Rahn is mainly interested in protein misfolding and metabolic disorders, and he accepted the invitation of the Association of Cell Biology and Differentiation to share his latest result on this topic. In particular, on May the 23rd, he held a lecture at the University of Milan focused on the redox and protein misfolding homeostasis in endoplasmic reticulum. So, good morning, Professor Rahn, and thank you for having accepted to give this interview for the members of the Association of Cell Biology and Differentiation. So, why did you become interested in protein misfolding?
Why did I become interested in protein misfolding? Well, I have to say that I historically, from my personal history, I became interested in a signaling problem, a rather obscure signaling problem, to try to understand how a transcription factor that I had discovered as a postdoc, now coming on 25 years ago, is induced by stress. And the search for the cognate stress, the signal that leads to the expression of this transcription factor, led me through a process of elimination to the unfolded protein response, which is a signal transduction pathway that informs the rest of the cell about the folding environment in the ER. So it was really through a curiosity-driven process of wanting to understand a very discrete problem that I came to be interested in a problem that's more general. So I think actually that many researchers, this is what stumble into problems, and then they realize that it's an interesting problem, rather than I think it's only the most clever that can look at the landscape of biology and say, oh, that's an interesting problem, I'll study it. That wasn't the case for me. But now I think that protein folding, misfolding, the response to it is clearly an important aspect of the biology of aging and a variety of diseases. So it's a justifiable subject for study.
Okay, and we would like to go a little bit more in detail in your research. So in which way do the molecular understanding of the unfolded protein response may impact on the treatment of patients, according to you?
Well, so my research doesn't directly address this question. I think that our level of understanding of the unfolded protein response, the signal transduction pathways, is really not deep enough to be able to predict what would work therapeutically. But this is really not so unusual. There are very few areas in biology where you can easily infer from the fundamental knowledge to where the therapeutic targets would lie. But I think it's reasonable to imagine that a deeper understanding of the way that a cell responds to a given stress can provide clues to therapeutics. And specifically, I think the way that this ends up working is that the homeostatic signaling pathways have a tendency to overshoot. So that for any complex adaptation to a stress, be it unfolded protein stress in the ER, genotoxic stress in the nucleus, whatever it is, the cell has a complex series of adaptations. And for any given situation, some of those strands will actually be enfeebling from the perspective of a biological outcome that we care about. And these enfeebling features of homeostatic responses are the ones that we target therapeutically. Right? We treat high blood pressure by enfeebling a normal mechanism that retains sodium that evolved in our history as a species when we lived on a diet that was very low in salt. And so that was an adaptive response. People that didn't have the ability to retain salt did very poorly. But nowadays, we target that pathway therapeutically. We enfeeble it in order to treat hypertension. So that's the analogy that I imagine would one day prevail, we would identify strands of the unfolded protein response that are bad for beta cell function or bad for some neurodegenerative disease, and target those selectively, right, to gain a therapeutic advantage.
So taking into account what you have just explained, which is the result that you consider the most important among the ones you achieved?
Well, so I cannot, in good faith or even in bad faith, say that anything that I have or that my lab has identified is clearly a therapeutic direction. That's a desire, right? So in the simplest world, what you would identify is you'd identify a signal transduction pathway. You'd understand how it evolved. You understand the advantages that it offered in evolutionary terms. And then you would understand the context in which it is enfeebling, and you'd be able to define the context in which you would want to inactivate it. And at several instances in our work, we thought that we had identified such a thing. So, for example, the way that eukaryotic cells respond to high levels of ER stress is by transiently attenuating protein synthesis. And in some circumstances, and this transient attenuation of protein synthesis evokes a feedback response that restores protein synthesis. So you have a kinase that inhibits protein synthesis and a phosphatase that restores protein synthesis. And in some circumstances, enfeebling the phosphatase actually makes cells more resistant to ER stress. It's as though the feedback loop is working too effectively for the given context. In this case, cells treated with tunicamycin. Now that, in theory, that could be a therapeutic target. You would say, well, if we have a drug that will weaken the phosphatase, maybe it'll make cells more resistant to ER stress. But the flip side is that the translational inhibition in other contexts is lethal to cells. So if you ask me today, if I had a drug that a drug that could inhibit the eif2 alpha kinase, would it be beneficial, and in what context? The answer is I don't know. But on the other hand, having drugs, I think this is a plug for drugs. The good thing about drugs, a drug that is titratable, so that you can have 10% inhibition and 20% inhibition and 50% inhibition, allows you to explore the therapeutic opportunities. So the process that I would propose would be identify a strand of a signal transduction pathway that has in some contexts a disadvantage, where we have an example of failure of homeostasis, because that's what it is, right? When enfeebling an arm of any response gives you a better outcome for the system that you're studying, you could say that's a failure of homeostasis. Identify failures of homeostasis, identify good drugs, compounds that are tunable, and then play around with the conditions. And of course, genetics just doesn't do that, because genetics is all or none. You either have the allele, well, you can do, maybe you have a few shots. If you have an allelic series, you can study it. But rarely do we have allelic series to enfeeble a pathway. We just have the wild type and the null in most cases.
During the lecture that you held in Milan, you presented some of your data to the members of the Association of Cell Biology and Differentiation. Could you please give us an overview of the main topic of the lecture?
So here I presented essentially two anecdotes relating to ER redox, that is the formation of disulfide bonds in the ER. And I especially chose that subject because here in Milan there are several groups that are prominent in this area, and I thought it would be interesting for me to get some feedback on our work. So you see, we scientists are extremely selfish. Even when we're invited to give a talk, we actually want to learn something from our colleagues. So the two stories that I presented are really quite esoteric and have nothing to do with the overall theme of the grand theme of proteostasis. It's a detail. So I became interested in how redox homeostasis, that is to say, the proper formation and dissolution of disulfide bonds in the ER, how is that affected by by perturbation of protein folding homeostasis. In other words, does ER stress change the redox significantly, or does it defend? Is the redox defended? And we found, and I think we're not the only ones, that remarkably protein thiol redox homeostasis is very, very well defended. So cells at extremes of unfolded protein stress can really cope quite well with maintaining their redox homeostasis. But we did find one circumstance in which redox homeostasis is altered in a measurable way, and that is when ER calcium is depleted. In my lecture, I presented some ideas about how that might come about. And the second story that I took on in this lecture is the role of glutathione, which is a tripeptide that is a very abundant tripeptide and which clearly can serve as a redox buffer in many systems, most notably in vitro reactions involved where people study thiol redox. And so it's been widely believed that glutathione is important as a redox buffer in the ER. And the prediction would be that if you somehow got rid of the glutathione in the ER, that would have dire consequences. Particularly, it would affect the ability of the ER to maintain a reduced pool of chaperones that reduce illegitimate disulfides. And so we devised a little trick for getting rid of glutathione in the ER. And I reported on the consequences of playing this little trick. And remarkably, very little happened. So the interesting result here is that in our hands, glutathione in the ER is not important. And so sometimes knowing that something is not important is, well, at the very least, it's interesting. Because if everybody else told you it's important and you find it's not important, then that's an interesting result. That's what I'd say is interesting for scientists.
So you deal essentially with basic research. Why do you think it is important, and why did you decide to shift to basic research, having you a medical education?
Well, so that's a loaded question. I have to say that my goal, to the extent that one has goals, but my goal was to marry my interest or my education, because it's not really an interest. We talk about having interests, but really what we have is a process, right? We go through life and we acquire stuff, and that stuff influences what we think. So at the end we have interests, but it's not that you start off by having an interest. I don't know anyone who woke up at the age of 18 and said, I want to understand how the Golgi works. That just doesn't happen. So in my case, I was educated as a medical doctor, and I stumbled through the wards and became interested in medical things, and my role models certainly were the great curers of disease, right? What I would like to do, what I wanted to do, was to discover something that's relevant to a disease process. The problem is that it's not easy to discover something that's relevant to a disease process. In fact, a lot of the discoveries that are directly relevant to disease processes tend to be somewhat trivial. So if you want to do something really more deeper, you have to do basic research. So let's just look at the stuff that's au courant today. We're talking about genomic medicine. Genomic medicine would never be around if it weren't for Fred Sanger, who figured out how to sequence DNA. So here we have a chemist, Fred Sanger, interested in an esoteric problem of how do you sequence DNA, developed dideoxy sequencing, and that's the basis of genomic medicine today. So once you are confronted with that kind of sobering realization, I realized that I might as well try to contribute to basic research in the hope that I would add something to the mix.
And why do you think a PhD student should join your research field, and which suggestion would you give him or her?
Well, I don't necessarily know that a PhD student should join my field, but if they decide to join my field, I think there's a good chance that they will, if they work hard enough and think hard enough, they'll eventually discover something interesting, or at least they'll see their competitors discovering something interesting, which is the definition of a good field, right? Because in a good field, either you'll discover something or somebody else is going to discover something, right? In a bad field, you're not going to discover anything and nobody else is going to discover anything. So that's my plug for for my field. I think it still has some life left in it, and it integrates cell biology, biophysics. You have to think about both general principles of signaling and homeostasis, and think about some details, some very small biophysical details.
So the time for this interview has almost gone, and I have a very last question for you. How do you manage, after so many years in this field, to find new motivations to go on?
I think I don't really, I just stumble along. I'm like everybody else, I show up at work. My job is to pull the lab forward, and that's what I try to do. And I think I'm not unusual in that. I think most people that run labs, that's what they're motivated by, is just to get through the day and to do your job, and that job entails trying to think of new ideas and new things. And indeed, it's not easy. It's not an easy thing, but it at least provides for some stimulation. Well, you know that from your own life as a researcher, right? There's boredom, punctuated by rare moments of excitement, sort of like the life of a soldier.
So we are at the end of our interview now, and let me thank you again for your time and for giving us intriguing comments and precious suggestions. And I hope to meet you again as a host of the Association of Cell Biology and Differentiation. Thank you again.
Thank you very much for allowing me to come here and visit. Thanks.